DNA replication begins with helicase unwinding and unzipping at the replication fork. RNA primase synthesizes and attaches RNA primers that are complementary to the given DNA sequence. For the leading strand, the RNA primase is only added once, while the DNA polymerase continuously adds on nucleotides in one direction. Since DNA polymerase can only add nucleotides in the 5' to 3' direction, for the lagging strand, these nucleotides are added in fragments called Okazaki fragments. When incorrect nucleotides are added, nucleases remove them. Finally, DNA ligase adds phosphate in the remaining gaps of the phosphate sugar backbone, essentially zipping up the DNA strands. Unlike DNA replication, transcription does not involve the use of RNA primers. Transcription begins with initiation, where the promoter region upstream of the DNA sequence to be transcribed is bound by the RNA polymerase. Then, elongation occurs, where RNA polymerase adds matching RNA nucleotides that are paired with complementary DNA nucleotides of the coding DNA strand. The RNA sugar phosphate backbone forms. The non-coding strand of the DNA is left alone. As RNA grows, the completed segment separates from the DNA template. Before the mRNA leaves the nucleus, the introns are spliced off, while the exons are recombined together. A 5' cap and a 3' poly A tail are added to prevent degradation.
The mRNA then moves out of the nucleus and into the cytoplasm. In the cytoplasm, a ribosome made up of rRNA or ribosomal RNA and proteins attach itself to the mRNA. A tRNA or transfer RNA carrying an amino acid then moves into the A site. The tRNA then moves on to the P site of the ribosome, while another tRNA moves into the A site. The carboxyl group in one amino acid reacts to the amino group in the other amino acid. This results in a peptide bond. The amino acids begin to form a polypeptide chain, and the first tRNA moves to the E site, where it is ejected from the ribosome without an amino acid. The A site tRNA then moves on to the P site, while another tRNA enters at the A site. Another peptide bond is formed between the amino acids, and the polypeptide chain lengthens. This continues until a stop codon is encountered. The ribosome then releases the mRNA and the polypeptide chain or protein into the cytoplasm.
Substitution mutation is when one nucleotide is swapped for another nucleotide. This can result in a different amino acid being coded for. The mutation can also be a silent mutation, which is a harmless mutation due to multiple triplets coding for the same amino acid. An inversion is where a number of bases reverse their order in the sequence. Depending on the size of the mutation, the amino acids can be completely reversed. A nonsense mutation is a point mutation that results in a stop codon. Frameshift mutations result from an insertion or deletion of a nucleotide from the sequence. This can be potentially very harmful, as it shifts the entire reading frame.